Então, dando prosseguimento ao nosso 8º Congresso do BRAIN, vamos seguir para a próxima palestra do dia, né? Então, é com grande satisfação que eu apresento o Dr. Diogo Veiga, que é pesquisador, jovem pesquisador da FAPESP, do programa Jovem Pesquisador da FAPESP, que fez seu doutorado no MD Anderson Cancer Center, lá da Universidade do Texas. Trabalhou como pós-doc na Universidade de Montreal, no Canadá, e está como jovem pesquisador na Faculdade de Ciências Médicas, trabalhando junto com o grupo da professora Ícia. Então, o Diogo vai apresentar para a gente a sua palestra sobre ferramentas de ômica e single cell aplicadas ao estudo do cérebro humano e das doenças neurológicas. Então, sem mais delongas, vou passar a palavra então para o Diogo. Muito obrigado, Diogo. Vamos lá.
Boa tarde, muito obrigado, professor André, obrigado pela comissão organizadora, pelo convite. Eu preparei a palestra em inglês, então eu vou mudar para inglês, se vocês não se importam. Poderia mostrar meus slides, por favor? The production team can please let me know if you can see the slides. Okay, so thank you everyone. It's a pleasure to be here on the first time for me at the BRAINN Congress. Thank you so much for an invitation. So I'll be talking about our work on the single cell omics and single cell methods to study the human brain, and also give you an overview of the field, the current state of the art of the field. Okay. So I'm assuming that everyone is seeing my slides fine, so keep going. So in the last 15 years or so, we have seen a great advance in our ability to study or to map the human genome and as well as the human epigenome. And as we know now, 98% of our DNA is no coding. It's not coding for genes, and it's collectively known as the regulatory genome. So nowadays we have several high throughput sequencing approaches for studying these non-coding regions. For instance, we have let me change my. We can study the chemical modifications of histones, several types of methylations, acetylations using ChIP-seq. We can study methylated DNA regions, and more recently, we can now start looking at the open chromatin regions of the genome that are accessible for transcription. So, as you know, most of our DNA is wrapped around nucleosomes, and therefore is not accessible for transcription, and not accessible by the transcription machinery. On the other hand, the open chromatin regions are those stretches or the regions of the DNA where the transcription factors and the polymerase can bind. So these include, of course, the promoters, but also include enhancers, silencers, and other regulatory elements. And the goal of chromatin analysis, open chromatin analysis, is to identify these regions across the genome, and to quantify these regions and to quantify the accessibility of these regions in different samples. So more and more we know that chromatin remodeling is highly dynamic and is associated to several human diseases, and understanding how these elements are affecting gene expression will be key to understand how how important genes are associated to disease. So there are several initiatives and groups working with the open chromatin. One is the ENCODE Consortium, which has recently have done a very true and systematic analysis of the human open chromatin in most tissues, including the brain. In this particular study, they use the DNA-seq experiments, and DNA-seq makes use of an enzyme that can fragment and specifically the open chromatin in the genome. So they identified around 3.5 million open chromatin regions in the human genome, and most of those regions are tissue specific. The other studies, including this one, have looked also in the human body across different tissues, and they come up with a very similar conclusion that the tissue specific the enhancers across the genome are not expressed across all tissue types, but rather they are specific to a certain set of cells or tissue type. So, for instance, you have the enhancers that are expressed only in the heart, only in the hematopoietic system, or only in the brain. Of course, when we look at the genes nearby these enhancers, they could find that those genes are associated to, as expected, to neuron differentiation or myelination, and this sort of baseline processes that are related to neurons and brain cells. Of course, the other application of this data is to look at the underlying sequences and to find what is the transcription factors that can be driving the expression of those enhancers. And then in the case of the brain specifically, they find some of the known transcription factors that control embryonic and adult neuron differentiation. So nowadays we have a couple of high throughput sequencing approaches for studying the chromatin, the open chromatin. It started with the DNA-seq and the FAIR-seq, but more recently with the development of the ATAC-seq, it really was a breakthrough in the field that popularized the high throughput genome-wide analysis of the chromatin in molecular biology and also in disease in human samples. So ATAC-seq is the assay for transposase accessible chromatin. So what it does is to capture these open regions of the chromatin. And it does that by, so the principle here is to incubate the nuclei of cells with the TN5 transposase, which is an enzyme that carries some adapters and will insert itself across the open chromatin regions. And doing so, it will also tag and fragment these regions. So everything that's not wrapped around nucleosomes can be then identified by these sequencing adapters that can then be amplified and later on sequenced in standard Illumina equipment. So there are ATAC-seq really there are, in contrast to other open chromatin experiments, ATAC-seq has two major advantages. One is that it needs a much lower starting material. The previous approaches, you needed an order of 10 to the 6, 10 to the 7 cells. So it was only possible doing experiments with cell lines or high amounts of tissue. And at ATAC-seq, you can do an experiment with a few thousand cells. And these, so enabling the analysis of clinical samples. Another point is that the preparation time is reduced drastically with this experiment. So it started. So the original protocol was introduced in 2013. Then there was some optimizations and some modifications that that were specifically for working with the blood cells and low quantity of blood cells, and also with other modifications to study frozen tissues from autopsies, for instance. So as I mentioned, so it's been a couple of years now, and ATAC-seq has been used in large-scale studies to map the open chromatin in humans in both healthy and disease state. So here are a couple of examples of major studies that used ATAC-seq for open chromatin analysis. So there was this includes hematopoietic system, blood, of course, the adult brain, and a very big study by the Cancer Genome Consortium, the TCGA, where they mapped the chromatin, the accessible chromatin of several human cancers, including glioblastomas. So there is quite a lot of information about in both in healthy and disease. So in my previous work, we have looked at open chromatin alterations in lupus, which is autoimmune disease, and in particularly in patients that have a severe form of lupus that with kidney inflammation. So we looked at the blood of these patients in CD4 T cells, and we looked using complementary approaches. So both ATAC-seq for the accessibility and RNA-seq for gene expression. And so we could identify the genes that are being modulated in disease, as well as the putative regulatory elements or enhancers that are controlling gene expression in the disease, in these patients. And so, long story short, we have we do have a lot of chromatin remodeling in this autoimmune disease, and I think one of the key messages was that there is for the majority of genes that are upregulated, that are increasing expression in the lupus patients, we could find the open chromatin regions nearby those genes that explain this upregulation. So there is a coupling of gene activation of transcription and the remodeling of and the chromatin remodeling. And here, for example, we found several cytokines and cytolytic molecules that are hallmarks of the disease, and we could find that they are overexpressed, but also we could find exactly what are the upstream enhancers and chromatin opening in the regions nearby those genes. So there's always a correlation between gene expression and chromatin remodeling. Another thing that you can look at when you look at chromatin open chromatin data is to identify the transcription factors that are associated with disease, that are driving the disease phenotype, and that would be through by doing a motif analysis that usually is called genome footprinting. And this motif analysis, motif enrichment analysis, will look at specifically at regions in the open chromatin regions that are, for example, being with a gain of accessibility in the disease. And then you can find what are you can rank transcription factors that are mostly implicated, that are binding to those regions. So it's really finding the protein DNA binding sites through this approach. So it's a quite interesting application of ATAC-seq. So parallel to this, there is a lot of development in single cell approaches, and single cell methods allow the sequencing of epigenomes and the transcriptomes of individual cells, rather than the bulk of the cells. So when you do a traditional sequencing analysis, what we usually call bulk analysis, what you get is an average expression of a gene in the tissue. So it's an aggregate signal. In the other hand, when you do single cell, what you're doing is getting the expression of the particular genes in individual cells, and then you can, of course, look at the expression in a certain cell type population. So there are several applications of single cell methods, and mostly the idea is to you can, for example, discover the cell types that compose the heterogeneous tissue, and also enumerate the cell types, and then look at the frequencies of the cell types when you compare these healthy and disease samples. Another application is to perform what's usually called to perform a cell differentiation analysis only based on the single cell data. Because you have thousands of cells, you can organize, and you can try to organize the pseudotemporal organization of the cell differentiation, for instance, and understand how the cells modify themselves through maturation. So if we go back to that figure that we had initially, the bulk open chromatin, we get an aggregate signal of the cells that compose the tissue. So we know that these particular enhancers and promoters are accessible, but that's an aggregate signal. With a single cell, you can then get a more detailed, refined analysis of how these open chromatin regions are accessible in individual cell types. So, for example, here you see a particular promoter that is open across all cell types, but this enhancer is specific to this cell type. So, this sort of analysis only if you can identify single cells. And so there's been a lot of development in the field in the last 15 years, or maybe something like that, 12, 13 years. There is now so the first methods was a proof of concept that could isolate one cell and sequence that cell individually. But the first methods were really in the order of dozens or hundreds of cells, and now we have commercial platforms that allow to sequence thousands of cells, or even tens of thousands, depending on the application or depending on the platform. So there was an exponential scaling the throughput of this equipment and these methodologies. So most of the single cell methods, they rely, of course, on the physical separation of cells and barcoding and the barcodes that will identify those cells. So, for instance, you have the FluidGAM platform. It's a microfluidics chip, and the droplet-based platforms such as the 10X and BioRad and also the microRel-based applications. So all these approaches require some equipment to perform this physical separation. There is also other approaches that use combinatorial indexing, and in this case, the nuclei or cells will be split into wells, and then they'll be tagged by a barcode. And this process of split and tagging is happening a couple of times, so that at the end of this split and tag, each cell will contain only one unique random barcode. That's another approach as well. So perhaps the most used platform is the 10X Genomics. That's probably with the SmartSeq, is together with SmartSeq and one of the most used platforms. And the idea here is physical separation of cells using a microfluidic equipment. And so usually you have 3 inputs, the cells or the nuclei, these barcode gel bits, which are specific tags that will mark the cells, and this partitioning oil, which is an emulsion oil. So when you load all these into the chips, you run your sample through the equipment, and as you run your nuclei will be the nuclei and the cells will be isolated in droplets, and these droplets ideally will contain one unique barcode that will identify that particular all the cDNA or all the RNA that contains that cell. So once the cells are separated in these droplets, then there is a process of reverse transcription, and then after reverse transcription, linear amplification, and then several other steps for the library preparation before sequencing. So this approach is quite popular, and it's been dominating the publications in the last high-profile publications in the last couple of years. And so once you've done all the analysis of the samples, and I have to mention that you have to have viable nuclei and viable cells to run through this approach. So once you have this data, once you sequence the data, you end up with thousands of cells, and these cells are essentially shallow sequenced genomes or transcriptomes, right? Depending on what you're doing. So these thousands of cells. So one of the most important application here is to do cell type discovery and characterization. So, for instance, the first step in the analysis is to do a pre-clustering of the data, and in this type of maps, each dot corresponds to a cell, and the nearby dots or have similar transcriptome profiles or epigenomic profiles. Then the next step is to do a cell type annotation. And there are several tools that can be used for this step, but usually this is one of the steps that you require the input of a domain expert to look to make sure that you have all the cells that your experiment is being able to identify all the cell types in the tissue, and this in this cell type annotation and this pre-clustering is done with no a priori information. This pre-clustering, I mean, is done with no a priori information of cell markers. So there is no need to know surface markers. You can identify grouping of cells or cell types only by clustering cells. And after this identifying the major cell types, you can then go to a more refined subclustering, which is taking one of the big clusters and breaking down into smaller populations. So, for example, in this Alzheimer's disease study, where they use the 10x genomics platform, they sequenced 80,000 cells for 48 individuals with different these levels, intensity of the disease, and so they find, as expected, the different types of excitatory neurons, inhibitory neurons, astrocytes, oligodendrocytes, microglia, and so on and so forth. Then they go into each of those major cell types and do additional clustering to identify subclustering, 13 types of neurons, excitatory neurons, 12 types of inhibitory neurons, and so on. So that, of course, opens a lot of questions and hypotheses to validate these in the tissue, right? And another important application is to look at how gene expression changes according to cell types. And so when you look at different cell types, you might expect when you have a bulk tissue, you just know if there's a general upregulation or downregulation. But here you can you can do a much more detailed analysis and then look at for example, at how gene expression changes according to different cell types. And here they found that while in neurons, both excitatory and inhibitory neurons, there is a downregulation of genes, you see upregulation in the other population, the other cell type compartments. So this type of information you cannot get from the bulk. Another application that we are actually very interested in is identifying enhancer gene interactions. And here, what is done is by correlation of the chromatin level of the enhancers with the gene expression level or the promoter accessibility. So when you correlate the nearby, by doing a correlation, you can identify if there is some sort of dependency. So for example, for this example, you can see here that this enhancer, this linked enhancer, is expressed in the cells 1 and 2 that have expression of the gene, but not in the cell types 3 and 4. So using this type of correlation, you can find putative enhancers for genes. Yet another type of information that you can get from the data is looking at overlapping the GWAS studies, the genome-wide association studies, the genetic variants that were found to be associated to disease with the single-cell data. And here is just to show an example that you have several genetic variants that are associated with disease. And then when you look at the expression of this in different cell types, you see that they are particularly overlapping the open chromatin of a given one of the cell types. So you can find whether you can probably hypothesize that these are involved in the disease. So, so there have been a lot of high-profile studies of the human brain using single-cell epigenomics, and for instance, this work, they have applied single-cell ataxic in 10 individuals to sequence around 70,000 cells, and of different regions of the cortex as well as of the hippocampus. And what they see is that basically with only epigenomic data, you can identify the entire diversity and heterogeneity of the brain tissue. You can reconstruct that with high accuracy. And another important message was that a lot of those open chromatin regions are specific to cell types. So they are expressed in a given cell type. They also look at the how they also found here that 3/4 of these SNPs, of the SNPs that associate to neurological diseases, are overlapping regions of open chromatin. So we know that most of the genome is non-coding, and 3/4 of these SNPs are indeed overlapping open chromatin and in specific cell types. And this changes from disease to disease. So for example, in Alzheimer's disease, SNPs are in the open chromatin of microglia. In epilepsy, you have an enrichment for oligodendrocytes and excitatory neurons, and in other diseases, you have different cell types. So this is quite a remarkable observation that might lead to different to new hypotheses about how these diseases are controlled. Another important study that was on the human in human prenatal at mid gestation. So they applied the single cell ataxy to study the cortical development. And in this case, they looked. They also have tens of thousands of cells, and here is probably one of the most complete prenatal open chromatin data available so far. So they look at different. They use the 10x platform, which is a platform that can scale up for this type of analysis, and also they can find the major cell types only with epigenomics data, and also to identify the regions of open chromatin across the entire spectrum of cell types in the brain. So one thing that they did, they went one step ahead, and they look at how because they sample different time points in the mid gestation, so they could also reconstruct or study the maturation or the differentiation of neurons, specifically into the excitatory neurons. So there are several transcription factors that control this regulation, and here they after you do you can get your epigenome, the epigenomes of the thousands of cells that you have, organize them by maturation status, and here you have the early progenitors, and here you have the more mature progenitors. And then you can infer the gene expression, or better, the transcription factor activity across these populations. And as expected, you see some SOX and HAS2, they're highly expressed in the early progenitors, and others transcription factors are associated to control of the later neurons. So it's a quite detailed analysis of how the cortical neurogenesis happen in the mid-gestation. So single cell can be done also in multiple modalities, and that means you can take the cells and run several, split the cells and run them through several approaches. But also there is this approach where you do matched joint expression of two modalities in the same cell type. And these are quite challenging and technically difficult, because you're dealing with a few picograms of RNA and DNA in each cell type, in each cell, individual cell. So you can do, let's say, gene expression and methylation, or gene expression in chromatin analysis in the same cell. These works are starting to pop out, so we're going to see more and more of this coming out in this year or so. And we, for instance, here it's they've used the multi-omics and single cell for studying the mouse brain and the frontal cortex and the hippocampus. So when they look at the chromatin, several histone modifications coupled with RNA, and chromatin activity coupled with the RNA. So this is like a completely entire reference, complete reference epigenomes for the cell types in the mouse. But perhaps one of the major studies from last year was that the release of the brain cell atlas of the motor cortex, where it was a group of researchers across several institutions that have looked very detailed on the primary cortex of 3 of the humans, monkeys, and the mouse, and using multiple types of single cell methods. And that includes single cell RNA sequencing, ATAC-seq, and also spatially resolved transcriptome analysis, especially single cell analysis, so this is an important resource for us working in the field, and has been available in a few months now. So now we also are launching a project on single cell that will apply single cell methods to study the childhood brain. And this project is a collaboration between the brain CEPID here with the Mac University, the Jewish hospital, and it's funded by the Chan Zuckerberg Initiative, and is also part of the pediatric networks, which is a subgroup of work at the Human Cell Atlas, working specifically with children. And what our project, so we will be doing joint chromatin analysis, chromatin and transcriptome analysis of pediatric brain tissue using the multi-ohm technology from 10x, which allows us to measure both chromatin and gene expression in the same cells. We have in total 18 pediatric samples that were selected for this work from 7 donors ages 3 to 15, several regions of the brain, and we will be looking at both normal and some lesion samples. So these are neurosurgical samples that were collected and analyzed by pathologists to be either a lesion or a normal tissue sample. And we are now validating, optimizing nuclear isolation and doing the library constructions for these samples. In the second year, we will be expanding this the analysis for the whole set of patients, and in the third year, we will be doing some integrative data analysis to understand a single cell landscape of the childhood brain. So this is a quite important project, because there's really a lack of reference to study normal childhood brain, and this affects the study of how neurological diseases are in children, right? So so here I think I have 1 minute left or something like that. I would like to thank all the team in Brazil and Canada, and our collaborators. So we have funding from FAPESP and funding from the CGI initiative. Okay, thank you for your time, and I'm open for questions.
Muito obrigado, Diogo. Muito obrigado pela apresentação. E nós temos aqui tempo para questões. Antes da gente, inclusive, entrar para as perguntas, uma informação importante para quem está acompanhando o Congresso, só reforçando que tem uma sessão de pôster às 16h10, né? Então, na plataforma, em algum momento, ficou sem ficou faltando, por uma confusão, ficou faltando esse dado, que agora tem a sessão de perguntas, vai ter um intervalo de 10 minutos, das 4 até as 4h10. Aí, às 4h10, vem uma sessão de apresentação oral da área de neurociência aplicada à clínica, tá? Então, fiquem atentos, que vai ter, como recebido por todos lá na programação encaminhada por, acho que, e-mail, fiquem atentos que às 16h10, então, tem sim essa parte dessa sessão oral, né? E também, relembrando todo mundo que vocês podem postar lá na plataforma as questões que eu vou repassando aqui para o Diogo, né? E, Diogo, enquanto o pessoal está organizando as questões, eu já vou aproveitar para fazer uma pergunta, né? Sobre a correlação, ou na hora de juntar, né, os dados de cromatina aberta com os transcritos, com o RNA-seq, como que seriam as estratégias para conciliar a descoberta de novas regiões codificantes? Porque, especialmente, vários tecidos se aplicam, mas no cérebro, especialmente, muitas vezes se encontram, quando faz o RNA-seq, principalmente quando você tem a possibilidade de ver populações celulares separadas, né, se encontram, especialmente, genes RNAs regulatórios, novos, não descritos, né, podem estar lá nos dados. E aí, imagino que já deva ter pipelines ou formas de incorporar, de conciliar esses novos transcritos com esses dados de cromatina aberta?
Sim, existem várias. É uma área ainda em desenvolvimento, né? Porque você tem os experimentos iniciais se faziam separado, a cromatina aberta e o transcriptoma das células únicas, né? Tem várias ferramentas, tem Snap Attack, tem o CRA, que é uma ferramenta que é tradicional na análise de single cell, que já tem módulos para análise, para integrar esses dados. Então, eu diria Snap Attack, CRA, e são as duas principais. E a ideia é sempre essa, né? É sempre tentar correlacionar a expressão porque você não sabe se nem tudo que você identifica no experimento de ataque-seq vai ser uma região reguladora. Na verdade, a metade dessas regiões talvez tenha um potencial regulador. E a ideia é para isso, ou você utiliza uma abordagem experimental, né? Seria um CRISPR, e aí testar individualmente uma mutação, essa região, ou você faz isso de forma computacional através da correlação entre a expressão, né, dessas regiões, a acessibilidade com a expressão do gene alvo, né? Então, basicamente é isso. Eu não sei se eu respondi a pergunta exatamente.
Sim. Era até pensando nisso e até pensando que você pode, imagino, filtrar, no caso do RNA-seq, para transcritos conhecidos, ou pode tentar descobrir novos transcritos. Eu imagino que pode acabar aparecendo isso também nos dados, né?
Eu diria assim, o problema da plataforma para esses transcritos não codificantes que é seleção poly-A, né? Então, tudo que você faz no RNA-seq com a plataforma 10x, ele tem que te expressar, ele tem que ser um transcrito com a cauda poly-A. Se não tiver cauda poly-A, não vai ser detectado. Então, e outro problema também é de genes não codificantes, de tentar encontrar a regulação de genes não codificantes, é que esses genes geralmente têm expressão mais baixa do que outros genes. E isso, então você tem que aumentar a tua cobertura na análise 10x, porque a análise 10x é uma análise, você sequencia 50 mil reads por célula. Então, o que você vai pegar ali é realmente o que está bem expresso. Então, tem essas duas questões que tem que ficar. Eu diria que se o objetivo é encontrar regulação de transcritos não codificantes, aí existem outras abordagens, como talvez Smart-seq, que é mais apropriada que a plataforma da 10x.
Legal. Aproveitando até um gancho nesse tema, o pessoal está organizando as perguntas ali, que você acabou até comentando agora, que cada célula, a profundidade é relativamente rasa, né? Uns 50 mil, 5 mil reads, sei lá. Mas, por outro lado, eu imagino que tecidos que são muito heterogêneos geram um problema de que você vai fragmentando. Inicialmente, você tem muitas células, 80 mil células. Mas se o tecido é muito heterogêneo, os subgrupos vão ficando com poucas células relativamente. Então, deve provavelmente ser um desafio isso de equilibrar quantas células você precisa capturar e sequenciar versus o quão esse tecido heterogêneo é. Aí, claro, está meio na infância essa aplicação no cérebro, mas isso é algo que já, eu imagino que a área já deve estar atacando, tentando, né, sistematizar esse problema. Isso é algo que realmente dificulta no caso de tecido nervoso, especialmente humano, que é super diverso, essa questão de ser muito heterogêneo, às vezes mais até do que esperado.
Eu acho que na verdade, para todos os tecidos humanos, não é só no cérebro, porque, na verdade, quanto mais você adiciona, mais sequencia, mais aumenta a cobertura, mais subpopulações você encontra. Porque às vezes você tem, e às vezes é difícil saber o que é uma célula, um tipo celular, e o que é um estado celular.
Funcional.
Um estado, às vezes tem um neurônio que está um pouco mais diferenciado, e aí parece uma população à parte, mas não é. Então, tem essas dificuldades também. Então, assim, é uma questão meio filosófica, eu diria até desse ponto, porque realmente ninguém sabe o que realmente tem ali, né? Mas quanto mais você sequencia, mais você tem possibilidade de identificar populações que têm a frequência baixa no tecido. Então, por exemplo, o pessoal do Human Cell Atlas agora, o que eles fazem é criar juntar os estudos de vários laboratórios e para que eles consigam identificar, talvez, populações que tenham menos frequência, né? Populações mais raras a partir disso. Mas então é sempre um trade-off ali, né, do que você vai, depende do quanto você vai gastar para fazer análise, né, e depende de quão expressa é a proteína que você também tem interesse. Às vezes você tem um interesse na proteína que está bem expressa, e aí você pode identificar se ela está expressa em todos os tipos de celulares ou não, né.
E uma curiosidade em relação ao planejamento dos projetos, né? Por exemplo, na iniciativa das amostras pediátricas, a ideia é criar esse atlas e também explorar essa diversidade celular, os tipos expressos. Você mencionou na palestra que, inclusive, cruzar as informações de DWOS, de sequenciamento também de diversidade de variantes, é super interessante. E aí, para caracterizações funcionais, assim, vocês têm ideia de que estratégias são factíveis, né? Porque eu imagino que em tecido humano, ainda mais pediátrico, obviamente, alguns tipos de abordagem são basicamente impossíveis. Mas no caso de quando encontra uma subpopulação nova, um mecanismo não descrito, a ideia é buscar localizar o que é fazer daí no à frente, no próximo passo.
Essa é uma ótima pergunta, né? Porque aí como uma validação funcional, seria o próximo passo que eu acho que todo mundo está tentando fazer, né? Então, uma vez que você tem os dados, aí você vai ter que desenhar experimentos específicos para validar se aquela variante genética está realmente modifica a composição celular do tecido, ou se aquela célula tem alguma contribuição para a doença. Então, eu acho que aí seria caso a caso para tentar entender quais são as abordagens experimentais, né? Tem tem uns estudos de CRISPR que eles fazem, e esses estudos de CRISPR pode ser que você perturba de regiões não codificantes, ou regiões de UAS, e aí a ideia realmente é saber se isso vai ter um fenótipo, por exemplo. Então, esses estudos também, eu acho que são importantes.
Legal, legal. Acho que depende muito do que encontra também, né? Tem uma
Exatamente.
pergunta que acabaram de postar aqui também, ó. Então, uma pergunta que veio da plataforma, da Maria Carolina Atier, se vocês pretendem integrar os dados de single cell com a localização das células, e se vai existir prioridade para o número de células ou a profundidade, o que vocês estão pensando em como equilibrar isso. Então, são duas perguntas. Uma é se vai ter alguma estratégia de localização, parte estrutural, e como que vai ser no projeto esse balanço entre profundidade e número de células.
Com certeza a gente está pensando em estender para transcriptoma espacial, né? Hoje em dia, eu acho que até a tecnologia permite você fazer resolver espacialmente a expressão para cada célula. Então, mas isso eu acho que tem também suas dificuldades, então depende da viabilidade do tecido que está armazenado, então tem uma série de questões que precisam ser verificadas. Geralmente trabalhando com tecido fresco é mais fácil para esse tipo de aplicação. Esse trade-off de número de células com a profundidade, eu diria que quando você começa, você sempre tem você procura, os primeiros experimentos é tentar identificar todos os tipos de célula que você tem no tecido. Então, se você faz um experimento e você não encontra os oligodendrócitos, então tem um problema, entendeu? Então, acho que a questão a gente vai começar com a recomendação standard de sequenciamento, que é de 50 mil reads por célula, e aí sequenciando 10 mil nuclei por 10 mil núcleos por amostra. Então, basicamente é isso. E aí também vai depender se aquele paciente, a gente vai conseguir aquele número de núcleos, né? Então basicamente é isso. Eu acho que a primeira ideia, eu não sei, assim, talvez tenha, como os experimentos são realmente muito caros, eu acho que não tem ninguém ainda fazendo esse tipo de, sequencia um pouco mais, um pouco menos, mas eu acho que a ideia é você ter um número, no momento que você conseguiu identificar todos os tipos de célula com uma profundidade de 50 mil reads por célula, aí você pode começar a fazer certas modificações no experimento. E lembrando também que a ATAC-seq é mais complicado que a RNA-seq, porque o ATAC-seq, o espaço a cromatina aberta é muito maior, o espaço de cobertura é muito maior do que a expressão de genes tem poly-A, né? Então, tem essa dificuldade. Então, a maioria dos dados de ATAC-seq são esparsos. Então, você identifica alguns picos em algumas células. Por isso que, na hora da análise, é importante você juntar por população. Na verdade, é o único jeito de fazer análise, porque hoje ainda não conseguimos fazer um sequenciamento que seja que consiga identificar toda a cromatina aberta a nível celular, né? É sempre a nível de população ou a nível de tecido.
Legal, muito bom. Vocês têm mais gente que tem perguntas para fazer, mas também qualquer pergunta que surja também pode ser encaminhado, mesmo na plataforma, pode ser repassada para o Diogo, e a gente sempre pode dar para encaminhar para a resposta posterior. E, mais uma vez, muito obrigado, Diogo. Eu acho que é uma área que vai trazer muita informação, com certeza, né? Fico imaginando que é uma área que vai ter uma corrida por profundidade também, porque, inicialmente, bom, a gente precisa achar os tipos celulares básicos. Na hora que eu acho os básicos, eles vão querer aprofundar para mais tipos e mais tipos. Vai ser bastante interessante acompanhar essa evolução dessa área. Muitíssimo obrigado, Diogo, mais uma vez. E reforçando, pessoal, um break agora às 4 e aí retorna às 4h10 para a sessão oral. Tá bom?